VEXAS syndrome, unraveling the mystery of overactive bone marrow stem cells. VEXAS syndrome, a perplexing inflammatory disease, has long puzzled medical professionals. Recent research from Memorial Sloan Kettering Cancer Center, MSK, has shed new light on its underlying mechanisms, offering hope for targeted treatments. The study, led by experts in immunology and blood disorders, reveals a fascinating interplay between bone marrow stem cells and immune responses. The discovery of VEXAS syndrome. VEXAS, vacuoles, E1 enzyme, X-linked, autoinflammatory, somatic, syndrome was first identified in 2020 by researchers at the National Institutes of Health. They discovered a unique mutation in the UBA1 gene, which is responsible for a range of severe inflammatory symptoms in patients. This mutation, occurring at position 41 on the UBA1 gene, replaces the normal methionine molecule with a different amino acid, leading to a cascade of health issues. The role of bone marrow stem cells. The MSK research team, led by Dr. Varun Narendra and Dr. Tandrila Das, uncovered two critical aspects of VEXAS syndrome. Firstly, the UBA1 gene mutation prompts blood-forming stem cells in the bone marrow to produce an excess of myeloid cells, a type of immune cell. Secondly, these myeloid cells' descendants, macrophages, become highly sensitive to danger signals, leading to self-destruction and the release of alarm signals. This self-destructive behavior may create a vicious cycle, attracting more defective immune cells and perpetuating inflammation. Unraveling the molecular circuitry. The study also delved into the molecular circuitry behind this overreaction. By using mouse models, the researchers demonstrated that blocking certain components of the death axis can reduce the inflammation cycle driving the disease. This discovery paves the way for potential therapeutic interventions. Challenges in studying VEXAS syndrome. The small and precise nature of the VEXAS mutation has made laboratory studies challenging. The MSK team employed a CRISPR-like tool called base editing to swap DNA letters at specific locations in both mouse and human cells, creating robust models to study the disease's mechanisms. Dr. Narendra, a specialist in leukemia and myelodysplastic syndromes, has been instrumental in applying this technology to hematopoietic cells, enabling the modeling of genetic variants in the lab. Synergy with immunology research. The study's findings align with Dr. Gitlin's immunology research, focusing on cell signaling and inflammation. Dr. Das and Lindsay Weyer-Suzuki, members of the Gitlin lab, conducted biochemical studies to understand why VEXAS mutated macrophages self-destruct in response to immune alarm signals like TNF and LPS. Their research revealed that these signals trigger macrophages to die in an inflammatory manner, propagating further inflammation. Implications and future directions. The study's insights not only shed light on VEXAS syndrome but also suggest that dysregulated cell death may be a more common cause of inflammatory diseases. Dr. Gitlin emphasizes the potential of these findings to lead to new therapeutic approaches for VEXAS syndrome and other inflammatory conditions. The research team's collaboration with other labs at MSK, including those of Dr. Caleb Leroux and Dr. Scott Lowe, has been instrumental in advancing our understanding of this complex syndrome.